Perfect. All right. Good afternoon, and I'm excited to present on a new project at EMSL called 1,000 Fungal Proteins. And as I dig into it, I'll do a very brief motivation and overview for the project, and then we'll go into touching on highlights from 2 of the pilot projects that we launched this year. And then I'll talk about how you can go about getting access to this capability going forward. I will point out that the 1,000 Fungal Proteins project is right now part of our decadal strategy. So this is something that will be around for quite a while. So with that, let's dive into the motivation. So 1,000 Fungal Proteins project is really something that's being launched in order to be able to characterize proteins from conserved fungal families that are largely uncharacterized either functionally or structurally or both. And this is really driven by the fact that our databases for genomes and proteomes have grown substantially, but functional annotation has not kept pace. And while structural annotation has had a huge revitalization through AlphaFold and other things, other types of computational modeling, there's still a lot of structural information out there that's missing. But regardless of if there is a high confidence structural model, functional annotation still needs to be done and be used to caught up. So the 1,000 Fungal Proteins project is kind of envisioned like a pipeline. It's one where we're really focused on user science and samples. And so it's been created in order to be able to be compatible with any types of samples that a user might provide. So from a user simply having a target gene or protein sequence that they might have identified through proteomics or other methods, we can start the entire pipeline through there. So a group does not have to have any cell biological or biochemical or even genetic skill sets in their group in order to be able to utilize the pipeline. Or you might have a target gene already in a expression vector, or have gone through the biochemistry in your group and generated a purified and isolated protein of interest. Or you may actually have a fungal strain that naturally has or has been designed to have a high copy number of whatever macromolecular complex you're interested in. So any one of these different types of pipelines basically are able to be fed into all of the structural and functional capabilities that we have here at EMSL. It's kind of drawn as a funnel right here because you've already heard about a range of different capabilities. Have over 150 high-end instruments on site at EMSL. So there's a wealth of information from nuclear magnetic resonance to cryo-EM for structural work to native mass spectrometry and other capabilities. So kind of all of those are wrapped up or described in this funnel, but really the output of whatever the user would provide is focused on functional screening, structural annotation, and post-translational modification profiling. This is really geared to link both the structural work and the functional work. For example, cryo-EM, most post-translational modifications are completely invisible. Even though we might solve the structure, we can't see what PTMs are there. And so the intent is to be able to do a complete deep dive into the characterization of the proteins, both structurally and functionally. Now, in order to basically create a rapid jump start to this project, we have a few core capabilities that kind of have been leveraged for the initial soft launch that's happened this year. And these will be the prime capabilities kind of forming the core of the 1,000 Fungal Proteins project into the next decade. So for individual protein structure determination, our Krios cryo-TEM plays a front role in that. We can either do microelectron diffraction of protein crystals, or if you have purified proteins, you can do single particle analysis in order to get atomic resolution without needing to do crystallization. But the Krios also has 2 other workflows that we're pulling in to the 1,000 Fungal Proteins project. One in case we wind up working with a protein that has either as a precursor or a product, a small molecule that has an unknown structure, or it's a new natural product and so forth, we have the ability to use small molecule microelectron diffraction to solve the structure of that small molecule or natural product and be able to identify the structure as well as chemistry that's ongoing when it interacts with the protein of interest. In addition, if we have a protein that forms a larger macromolecular complex, or you're interested in understanding the details of its localization or its interactions inside of the natural cell environment, we have the ability to do whole cell electron tomography on it and see every single density inside the cell unambiguously, and be able to identify the overall interactions that are occurring. And ultimately, if we have a high enough copy number, you could do subtomogram averaging and still get near atomic resolution of your protein of interest. So cryo-TEM is one of the core capabilities, but another core capability is the cell-free pipeline that we developed about 5 years ago. This allows us to start with that gene or protein sequence at the very beginning. As I mentioned, a user could provide just the uniprot sequence of your protein of interest. We get that into a clone compatible with our cell-free pipeline, and then we can do expression, purification, and then scale it up for either functional or structural analysis. And here, this pipeline currently utilizes either wheat germ, E. coli, or human cell lysates. But all of these, we have 4 different scales. And the smallest scale happens at like the 25 microliter scale, where we can test just for if the protein expresses, if it's in the soluble fraction, whether it's a membrane protein or soluble protein. If you need additives to be able to get it to fold properly or to be in the fraction that you care about. And then once we know the overall yield and that it does express under certain conditions, then we scale up for the different downstream approaches that we're going to use. Now, one thing to note here is the fastest time frame from actually solving, from receiving a custom clone of a protein to having the final structure only took 9 days. It actually took 27 days to get the custom clone from GenScript and 9 days to go all the way through this process, scale up, and then solve a cryo-EM structure for the protein to 2.5 angstroms. And this was the first ever full-length structure for this particular protein. That's not the standard for most proteins that we get with this pipeline, but it does highlight the ability to take just a gene sequence and then get overall structure and functional work out of it. Another thing, as I mentioned, we have many capabilities at EMSL. Because of that, we have the ability to do multimodal analysis and get additional information that we can't pull out just from a single technique. This is highlighting a capability where we combine cell-free expression, cryo-EM, and native mass spec to pull out information about a heterocomplex assembly that was kind of invisible to all the approaches on their own. So here you can see in yellow a pseudoenzyme. This is a protein that naturally forms into dodecameric barrel. But this is a protein that is an inactive form of PDX 1.3, which is an active enzyme. The two of them are so similar, they only differ by 200 Daltons at the monomeric level. But the previous model about how this yellow pseudoenzyme regulated the activity of this pink enzyme in cell was that it was through an interaction of preformed hexameric rings of both type, the pseudoenzyme and the enzyme coming together, and that would basically have the overall activity that the larger complex could do. We didn't believe that this was true, so we used a combination of cell-free expression where we did co-expression by including the two different types of protein, the vectors containing them at different ratios. We're able to show that we could create a range of complexes of various size, which automatically suggested this was not the case, but we wanted additional evidence. used native mass spectrometry to wind up showing that we could identify the masses of each individual stoichiometric complex. And with cryo-EM, we're able to wrap up the story and show that all of them actually kind of funnel together to the same answer of we're able to actually form every single complex from a 12 and 0 to 0 and 12 with every different stoichiometric ratio in between. And so what this is actually now showing is it's completely reversing the previous model that had been accepted by the community for about a decade, and is showing that the two of them interact dynamically inside the cell in order to regulate vitamin B6 biosynthesis. So this kind of just highlights, hopefully, some of the strengths of the capabilities we have to really do a deep profiling into the functional characterization, not only just at the single monomer protein level, but also how it interacts inside the cell environment, even within heterocomplexes. So this year, I mentioned we launched a few pilot projects. We launched about 6 different projects, including one where we worked with JGI's microcosm database to identify a few conserved gene families that were not functionally or structurally characterized. We started with those, but the next 2 examples are kind of the ones that were most successful and already have generated manuscripts from them. So in partnership with Jiwei Zhang, an EMSL user, we wound up having a series of protein targets that he had identified from proteomics work, where he believed these particular proteins, although they were unannotated, he believed that they were related to the degradation of lignocellulose. And so he took those sequences, sent them to us. We did cell-free expression on them. One of them happens to be a membrane protein. The others are all soluble proteins. But using computational analysis, predicted functions were identified. After expression, we wound up subjecting them to those functional assays and controls, and we're able to unambiguously identify and annotate functions for 4 of these proteins, all of them related to lignocellulose degradation. So this is a fantastic story about being able to start from gene sequences where limited information is present and be able to actually confirm that functionality. Taking it a step further, working with John Magnuson, he had a strain of fungi that he had comparative proteomics, comparing wild type strain versus a strain where he knocked out this CAD-A protein that's shown with the little red X. This is a protein that is cis-aconitic acid decarboxylase, which would normally would normally convert cis-aconitic acid into itaconic acid. Now, by knocking that out, it got a strain that overexpressed cis-aconitic acid and secreted it into solution. And cis-aconitic acid is actually really highly relevant for industrial purposes, for plasticizers and other biopolymers. And so wanted to identify how this strain was not only producing it, but also exporting the cis-aconitic acid. Through that comparative proteomics, they identified a membrane protein that they believed must be involved with the export of cis-aconitic acid. And so what we did is take this protein, use cell-free expression directly into proteoliposomes, and then we created a fluorogenic activity assay where in the absence of any acid in solution, in the buffer solution, the proteoliposomes would be completely blank. You would not see any signal. But as soon as an acid was in the outer solution that was able to be transported by this protein to the inside of the proteoliposome would acidify and fluorescence would increase. And so lo and behold, when you add cis-aconitic acid, you get a very strong signal from fluorescence showing that it's very specific for transport. Itaconic acid, which is very structurally similar, tricarboxylic acid, shows a very weak signal here. It's about 15 times less intense, suggesting that there is some leakiness to it, but overall you have a very high specificity for cis-aconitic acid. two other tricarboxylic acids that are very structurally similar, 3-HPA and citric acid, showed absolutely no detectable signal and no transport. To confirm that this result wasn't actually just the result of decay of the fluorophore over time, at the end of the experiment, we reran cis-aconitic acid and showed that the fluorophore was still completely viable. And so this was able to do true functional annotation, identify this protein called AEXA as a cis-aconitic acid transporter. We were further able to go on and determine its overall atomic structure. So wrapping together both from an unknown gene that was previously unannotated, being able to annotate it both functionally and structurally. So this is kind of the focus of what the 1,000 Fungal Proteins Project is going to be all about. And so I don't have time to go into all the details, but in the question of how to access it, you as a user can apply to any of the normal EMSL calls. The 1,000 Fungal Proteins Project, because it is a strategic focus for the next decade, it is a topic of high interest for all of the FY24 calls and will be for FY25. We have created a separate website up at the top that provides additional information and contacts for as well for myself and Scott Baker, so that you can contact either of us co-leads with it to get more information. But really the focus of all of this is trying to engage the community as much as possible to provide targets for us in order for us to help do functional and structural characterization to further advance your science. So we're really focused on user-driven protein targets that come in from user calls. As I mentioned, we're working with JGI microcosm database in order to solve or characterize proteins from the conserved families of unknown function. But in later years, as we ramp up the automation and overall productivity, we're also planning to engage larger consortia projects where we pull in multiple investigators from multiple universities to tackle really large challenges like fungal regulation networks and so forth. Finally, we really do believe that the 1,000 Fungal Proteins Project, one of the best ways to get engagement is to help get community involvement. And so we are planning FY25, 2025 summer school will be focused on 1,000 fungal proteins. Every student that comes and joins that will get direct hands-on experience with every step of the overall pipeline, so that they can understand kind of the overall workflow, but also the benefits and potentially limitations related to their proteins of interest. So with that, I'm happy to take any questions.
I see a question. Okay, thank you.
Very nice talk. I was just interested in how do you control the orientation of the transporter? You know,
That's a great question. So some proteins will have a non-specific orientation within it. In this case, the only way that you would get acidification on the inside is if you have transport to the inside. It is possible, but in this case, the organelle that we believe this protein is actually naturally expressed in has a very high curvature to it. So we used very small proteoliposomes in order to mimic that curvature. So we believe that the curvature itself favored a particular orientation. But it is something that we would be able to confirm by cryo-EM if the protein actually had a larger cytoplasmic domain. This particular one is pretty much all transmembrane helices, so it would be hard to confirm.
I see. Okay, thanks. So that was John's retirement present, I guess. The other question I had was, do you use commercial kits for the V-germ and human lysate, or?
So we use commercial kits with some modifications in-house that have proven to be more reproducible than just the standard procedures, but yes.
Okay.
Hi, very nice talk. I was wondering, so your charge is obviously fungal proteins, but what is, if as a user you have a non-fungal protein you're interested in characterizing?
Yeah, so it's a great question. All of these capabilities are also available to anybody else. So the cryo-EM, NMR, cell-free expression, native mass spec, and so forth, everything that I talked about here, anybody can request based on their user project. 1,000 fungal proteins is, we have a larger strategic goal called DigiFen, and that is kind of trying to do a much larger scale focus on pretty much most types of BER relevant science. 1,000 fungal proteins is kind of a smaller version of DigiFen, trying to demonstrate that on a very focused or more focused target. So that's why we chose fungal proteins specifically. So in terms of the project, it's to demonstrate that smaller scale DigiFen. And while it does get a little bit of a boost because it's a high topic area of interest during the calls, we do the same types of approaches for plant, algal, bacterial,